being made available as we go, so I hope this will be a useful resource for as many people as possible. The next talk, we have a double-headed show with Andy Singleton and Kajsa Brolin. Where are you? You're here. We're going to tell us about GP2.
Thanks so much. There we go. Thanks. So it's a real pleasure to be here. Thanks for the invite. It's also a huge pleasure to talk about GP2. I absolutely love this project. This is a project that has been going on for about 4 years. I co-lead it with Cornelis Blauendraat, who's sitting all the way in the back. I love this project for many, many reasons. It's about doing research at scale, it's about doing genetics at scale, but it's also about enabling people from around the world and finding the kind of next generation of leaders. So it's great to do this talk with Kajsa, and we get to find really, really talented new generation of researchers and get to work with them, and Kajsa is one of those. So this broadly is what we're doing in GP2. It's a genetics initiative, so our aim is to collect probably somewhere in the region of 250,000 people from around the world and understand the genetic basis of disease in those individuals. There are some key tenets for this work around democratization of data, so not just making data available, but giving people the resources and the skills with which to analyze the data. Really focusing on collaboration, cooperation, sharing data in a safe and responsible way, which is a huge challenge as laws and regulations change around the world. You can imagine that takes quite a lot of effort. Ensuring that there's diversity in the research subjects that we work with, but also the researchers we work with. Again, the idea here is to create a group of researchers around the world who are working collectively to make new discoveries, and ultimately to create resources that are actionable, right? We're not doing genetics just to find new genes. We're doing genetics in order to try and find therapeutics. So our mission, I've really said this, to dramatically expand our understanding of the genetic basis of Parkinson's disease and to make that knowledge globally relevant. Clearly, if we think genetics is an initiating point for coming up with viable therapeutic options, then you need to understand genetics in populations around the world. The path, I would say traditionally what's happened is that really well-funded labs have gone into underrepresented populations, worked with a local clinician, grabbed samples, brought them back to their lab, published, that's it. This is different in that we aim to create a global research community. So really train folks who've perhaps not had the opportunity to learn about genetics and not had the opportunity to really get involved in research. Train them how to set up research, train them how to analyze data, train them in every aspect of this work. The resource, again, generating deep genetic data on 200 to 250,000 individuals. And we do this through really this structure here, global collaboration and capacity building, so building research capacity through skills and sometimes physical resources around the world, growing the next generation of leaders that can continue this work moving forward. Applying transformative genetic methods, so the things you would expect, like deep genotyping, whole genome sequencing, long read genotyping and long read sequencing, and then accelerating discovery by democratizing data. And again, I think in genetics we've been very good at just making data available. This is more than that. This is making data available, making compute resources available, and teaching people how to actually apply those compute resources. And the idea here is to create this self-propagating discovery engine. I think GP2 will have been a failure if there's just a bunch of publications coming from my lab. It'll be a success if there are publications coming from all over the world. And that seems to be what's happening so far. I show this just to say there are literally thousands of people involved, thousands of researchers involved in GP2. I get to stand up here often and give these talks, but really there are an enormous number of people doing a huge amount of work to make sure that GP2 functions successfully. This is roughly where we are. We're about 4 years in. really heavily into the production phase. You'll hear a bit about data production right now, but things are going really well. Obviously, we started this global initiative during a global pandemic, but that really gave us a lot of time to set things up and make sure that operationally we were really in a good place. So with that, I will hand this over to Kajsa. Kajsa.
Thank you. Yes, so just to show where we are right now in GP2, so we're generating data from studies across the world, and currently we have, or in the latest data release, we had 55,000 samples from 60 unique locations all around the world, and we are expecting around 180,000 samples currently, so I would say we're on the track of hitting the 200,000 at least. And if you want to see more about the sites, you can scan this QR code. I hope it works. Also to say, since we're harmonizing data from different cohorts, to mention the value aspects of contributing cohorts, and they benefit from a lot of things, including data access, authorships, and international collaboration. That's really what GP2 is about. So in regards with the samples, we provide genotyping of the samples, and we're also transitioning now to sequencing. And the data that GP2 generates, so the clinical data and the quality-controlled genetic data, will be returned to you. And the contributing cohorts also have access to the GP2 analysis working group, which is a great group of knowledgeable people and also for free computational resources and input on your studies and research. And of course, you get to publish your own data and also have access to the entire GP2 data set. And as I said before, authorship on publications. And what I really think is fantastic with GP2 is the fact that it's a very collaborative global network with a lot of opportunities. You have the networks of top geneticists like Andy, and also, there's a lot of training opportunities as well for junior scientists. So it's a lot of benefits for joining GP2. So just to see where we kind of are right now with what has been published in GP2, there's a lot of things ongoing, and here you just see two kind of major milestones. One of them is a multi-ancestry genome-wide association, a meta-analysis of Parkinson that included 4 different ancestral populations to kind of identify SNPs or genetic variants that are shared between these groups. And I also want to highlight this paper as well, because it's probably of more interest to all of you, and we heard a really nice talk yesterday and also a very nice talk today about it. So it is the identification of a new genetic variant in GBA in individuals of African and African admixed ancestry. So you heard this before, but repetition is always good. So this is a project led by Cornelis, Blauendrat, and other people in GP2, so it's also a very collaborative effort. And then a new GBA genetic risk variant was identified, which is almost completely absent in other ancestry populations. And this is a unique variant since it is non-coding, common, and high risk, which is not what we've seen for the other GBA variants in European populations, for example. And it confers, as I said, quite a high risk, 1.4 times higher risk in heterozygous carriers, and a 3.5 times higher risk in homozygous carriers. And it's very common, as we also heard before. And with this, it has a much higher population liability than other GBA1 variants for this population. And I also want to mention then that it is associated as well with a higher younger age of onset. So each allele results in approximately 3 years younger age of onset. And we also heard a bit about the function. So this is, I realized that this is Cornelis' paper and not the previous one, even though it's probably been on it, sorry for that. Yes, so since it is an intronic variant, it's of course interesting to know the mechanisms of this variant. And as we have heard, that it is a branch, it's an intronic variant that acts at the branch point sites, which leads then to this missplicing and misprocessing of RNA. So there seems to be an increased RNA transcripts with this GBA risk variant, but they seem to be non-functional, which then leads to a decrease in GBA proteins and also less GCase activity for the risk variant. So nice work. I want to mention some possibilities to study GBA1 and GP2. So as I said, we have a lot of samples and more coming in. So these are just some numbers that I put together looking at risk variants in GBA1 and the available data right now. And also want to mention here that more data is coming in. We have several cohorts that are included or will be included into GP2 that kind of focus on GBA1 and that are actively recruiting, so the numbers will increase further. And we're expecting that the numbers will increase even more since GP2 now has expanded across the PD spectrum to also include atypical Parkinson and also individuals at risk or that have a prodromal Parkinson. And this kind of goes in line with the entire aim of GP2 to examine genetic forms of Parkinson's, so even the other forms than the common Parkinson's disease diagnosis. So including both manifesting and non-manifesting carriers, such as non-manifesting GBA1 carriers. So there's many reasons, as I said, why this is in line with GP2 and why it's important to study, because we don't know so much about the genetics of early Parkinson, and there's also, the understanding of it in another underrepresented population is severely lacking, so it's really important. And we also do believe that genetic stratification will be valuable, if not even mandatory, in future clinical trials in individuals at risk then. And as I said, we are expecting the number of GBA1 carriers in GP2 will increase with this effort because one of the groups that we're interested in is then those at high genetic risk, such as GBA1 carriers. And this is just to show these are what we generally call prodromal samples, and we have around 5,000 now where all agreements are completed that will hopefully enter GP2 very soon. Just to also highlight some of the ongoing projects, we have heard about 2 of them before, and I think the QR codes work if you want to have a further look at them. So investigating this new genetic risk variants in both REM sleep behavior disorder and then also the mechanistic study of it. So there are a few more interesting projects ongoing, including looking at the genetics of progression. We also want to test the accuracy of DBA calls. We're using the NeuroBooster array for genotyping, and we're further investigating GBA in around 5,000 PD cases using nested PCR and long-read sequencing. And we're also looking at the penetrance of GBA. We're also looking at GBA in population from Malaysia, and then we have some ongoing projects to look at biomarker assessment and trial readiness, and also looking at potential precision medicine initiatives in GP2. So all of these kind of includes GBA. And love to talk more about these if you have any questions. So with that, I just want to, thank you all for listening, but also acknowledge the many fantastic people working with GP2. It truly is a highly collaborative effort, and this is not even all of us, but a small part. It's a really nice group. Feel free to email or visit our website. So thank you.
If there's any question about GP2, now is the time, and then we'll move to a panel. So, I will tell you about it later.